apoptosis or programmed cell death. Now, I will be providing the latest insights about this topic and myself, Dr. Ashfaq, Professor and Head, Department of Anatomy, SKIMS. So, as far as apoptosis is concerned, we will be dealing with the latest insights, the histopathology behind the apoptosis, and the therapeutic uses of apoptosis as far as its clinical relevance is concerned. So, going to the basics of apoptosis, as we are well aware that it is also known as PCD, programmed cell death, and it happens to be a very precise, highly regulated process in which the total count of the cells in the body is maintained by an intricate balance of cell proliferation and cell death. And this process is particularly useful in removing unwanted cells or preventing excessive proliferation of cells. So, this balance of cells is very important, and this is vital for the normal homeostasis of the body functions as well as inflammation and the development in case of multicellular organisms, ranging from the process of embryogenesis to all the processes involving immune regulation and degeneration. So, as far as apoptosis is concerned, what distinguishes apoptosis from normal inflammation or necrosis? So, as is evident that there is the cell shrinkage, it is very important. That means the cell decreases in size, and along with the decrease in cell size, what happens to the organelles packed within the cell? They get condensed. So, there is close packing of organelles, and in associated to that there is chromophilia. There is the cytoplasmic chromophilia, increased capacity for dyes. In addition to that, the chromatin condensation, which is a very, very vital feature of apoptosis. The nuclear chromatin aggregates itself into dense filaments. Now, as far as the regulation is concerned, you are well aware of the fact that there are factors everywhere, pro and anti, and the factors which are pro-apoptotic, and there are factors which are anti-apoptotic. So, as far as pro-apoptotic factors are concerned, you are well aware of APAF1, which drives the apoptosis, and BCL2, which is anti-apoptotic. Now, as far as the other factors are concerned, pro-apoptotic and anti-apoptotic, you have the P53 gene, which is the guardian of the genome and which is present on chromosome 17. And what is important, it is a very important pro-apoptotic factor along with cytochrome C and CA-NMC11, which I have not mentioned over here, but you have to mention the pro-apoptotic factors as well as the anti-apoptotic factors like the MCL1, BCLW, and BCLB. So they are very important anti-apoptotic factors. So the regulation is at a genetic level, and that's very important. And how do we detect apoptosis as far as pathology is concerned? So the DNA gets fragmented, and DNA fragmentation analysis is one of the important markers of apoptosis, in addition to annexin 5 assays. So annexin assays are very important, as well as caspase enzyme activity. So these three, DNA fragmentation, annexin 5, and caspase activity, determine or the lab determinants of apoptosis. Now, once we have studied this process, which can be physiological, which can go wrong at any point of time, so what are the therapeutic potentials? Basically, before understanding the therapeutic potentials, you have to remember that there is this balance, a very intricate balance between cellular proliferation and cellular death. And this is maintained, it is programmed, it is genetically programmed. But once there are abnormal, there's an imbalance between these two things, we can get excess of apoptosis leading to degeneration, or we can get decreased apoptosis which predisposes to cancers. So benign or malignant conditions which are there are a result of decreased apoptosis, there is increased cellular proliferation, be it the colonic malignancies, be it the gastric malignancies, be it the hepatic malignancies, the brain malignancies, the malignancies of the GIT unit. There's a fundamental process that there is decreased apoptosis, as a result of which there is proliferation of the cells and there's a higher malignant potential. And as a result of excessive apoptosis, what happens? There is more cell death, and that's the basic cause of neurodegenerative diseases, which are the modern pandemics like Alzheimer's disease and Parkinson's disease. So that's important. So this is to understand the therapeutic potential lies within the regulation of apoptosis. Now, as far as these malignancies are concerned, oncology, medical oncology, surgical oncology, it is on the rise. There are so much of a malignant potential, which is increasing. The number of cases of malignancies are increasing. The incidence prevalence of malignancies is increasing. The budget of the countries towards the allocation towards the oncologies is increasing, and it is becoming a very big issue with the advent of the treatments which are very, very costly. So what we can do, we can just have a very high impact factor as far as apoptosis is concerned. So what is important in the cancer cells is that they devise mechanisms to evade apoptosis, and uncontrolled cell death occurs. So targeting this evasion capacity of the malignant cells is very important. So you have to have, a regulation. So you can be therapeutically, this is a therapeutic challenge with the scientists. So cancer treatments focus on targeting apoptosis evasion. So you see that these cancer cells are smart and they find ways to evade apoptosis. So that is why you block that cycle. That's important. Then another thing I mentioned just now, BCL2 and anti-apoptotic factors and all those things. So you have to potentiate, some of the factors which are effectively, and there are certain molecules which target the cancer cells specifically. So potentiating these factors, increasing the pathways to effectively target cancer cells or the factors which target the cancer cell death, and just improving and increasing the viability coefficient of these factors, that helps in prevention of tumors and has got a very important therapeutic role. So in this context, we have got venetoclax, which is important in inducing cancer cell death. So we can potentiate the cancer cell death as well. Now, as far as P53 is concerned, it is called as the guardian of genome because it is one gene which is present on chromosome 17, and in more than 50% of the tumors, what happens? The mutations occur in this P53 gene, and this is very, very important. And reactivating P53 induced apoptosis in tumor cells effectively. So we can target, we can detect the mutations in the P53 gene, and they help. And this entity, the Lee-Froman syndrome, in which you have got a patient who has got a potential to have multiple malignancies because of the mutation of a single gene, P53. It's a very important thing, and we have to have this P53 in mind once targeting for cancer chemotherapy. Now, trial activation, this is very important. This is something about the surface. So that means these are cell surface proteins. What are trial receptors? These are cell surface proteins. And what is important? That they cause death in cancer cells while at the same time having no effect on the normal cells. So trial is important. The trial activation is important. Trial receptor agonists selectively induce death in cancer cells. So that is the objective. Now the malignancy is there, and you have to induce death in these malignant cells. So what we do trial activation. Though this is a separate topic, I will deal with it hopefully sometime in some other lecture in detail. But over here, you have to remember that trial activation is one of the important therapeutic modalities of inducing cancer cell death. That's important. Now, this was about the malignancies and all, other oncological conditions. Now, what is important that excess of apoptosis can cause increased cell death, and that's very problematic as far as especially the neurological conditions are concerned. We are well aware of Alzheimer's dementia, Alzheimer's disease, and we are well aware of the Parkinson's disease. In these diseases we can control the neurodegeneration, we can control the excessive apoptosis. So that is very, very important. So by targeting the neuronal apoptosis, which causes significant cell death, significant cell loss, and we can just prevent it at a very early stage and focuses on these things. So neuroprotection is important. Caspase inhibition, caspase enzymes, and caspase inhibitions are very important. They block the executional enzymes to protect neurons. So over here we have to see how the neurons are protective in addition to, I must say, antioxidant factors, the other factors which help in neuroprotection to a limited extent. So caspase inhibitors are very important, and I mean to say any factor which supports neuronal viability and prevents apoptosis is very important in the treatment of these diseases. Now, in addition to that, autoimmune diseases like the SLE, like the MCTD, and many conditions, therein the targeting the harmful cells and preventing autoimmune destruction is very important. So as you can see that the summary of therapies is that there's got a apoptosis got a role in cancers, neurodegeneration, autoimmunity. So these are some of the things which are there, and you are welcome to any queries. Thanks a lot.